Hello, everyone, and welcome to today's webinar titled Introducing ALiCE, Simple, Scalable, Cell-Free Expression to Difficult-to-Express Proteins. This webinar is a part of the 5th annual event in the Bioprocessing Virtual Event Series. I am Shelley Newlock of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by LenioBio. For more information about LenioBio, go to leniobio.com. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract window below the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Charles Williams, Head of Systems Innovation, LenioBio. For a complete biography of our speakers, please visit the Presenter tab from the menu at the left of your screen. Dr. Williams, you may now begin your presentation.
So thank you, Shelley, for the introduction, and to the LabRoots team for organizing this great event, especially this emerging expression systems track that is so relevant to our work at LenioBio. As stated, I'm going to introduce our system to you, our ALiCE system, which is all about cell-free expression of difficult-to-express proteins. A little bit about myself as your presenter. So I've been working in the LenioBio R&D department for over a year and a half now, working to express all kinds of different proteins in our system. And to understand a little bit more about our system and what we can do with ALiCE, it's also very relevant to understand where the company was founded and the origin story. So you'll probably remember back in 2015, there was this awful Ebola epidemic in West Africa, where people were dying every single day from this disease. At the time, there was a really huge news story when they released this experimental drug called ZMapp, which kind of hit the headlines as a plantibody drug, antibodies that were made in plant system. But actually, even though it gained global kind of fame at the time, there were only enough amount of this protein drug for 7 doses. So really not a very clinically significant clinical trial. And with some calculations, we landed on these numbers of 40,000 square meters of greenhouse, and 90 days would have been needed to produce any kind of useful amount of dosage of this system. And around this time is when our founder came across a related kind of expression system, so a cell-free lysate based on tobacco root cells, suspension root cell culture. And at the time, it's used mostly as a research tool, but it can produce proteins in 24 hours rather than in the months required from using a whole plant. So really taking this invention from the academic setting towards commercialization was the whole idea behind LenioBio, which was kind of framed by the central thesis of what if you could produce your protein in a matter of days and at different scales. So then that's where this tobacco BY2 cell lysate was termed as ALiCE and commercialized and developed for scalable protein production. So ALiCE stands for almost living cell-free expression. And really what we do is we take a suspension culture of these BY2 cells, we process them and strip out the elements that you really don't want when you're expressing proteins, things like the cell wall, things like the vacuole. And from that raw cell lysate, we add a couple small molecules as our master mix to complete it. And then just by adding DNA, template DNA that encodes your protein of interest, within 24 hours of a simple reaction, you can generate protein material for all kinds of purposes. So on screen, you see some examples of antibodies and growth factors and enzymes. You can think of these as kind of therapeutic candidates. But we've also produced numerous antigens and virus-like particles with more relevance towards vaccines or diagnostic purposes, and also some peptides. So to dive in a little bit further on what actually is the cell-free protein synthesis in general. So at the very heart, we're doing coupled cell-free biosynthesis. So you're coupling transcription to translation. Transcription normally driven by the T7 RNA polymerase in the literature, which takes your template DNA to produce the mRNA. And then you have the translational machinery to produce that protein. So in many cases, this translational machinery comes from a lysate of cells. So in the first instance, we have some upstream cell culturing production, which you then strip away the membrane, the cell wall, to produce the lysate. You have a reaction mix of some small molecules, the T7 RNA polymerase, as I just mentioned. And then within that one pot, you simply add that DNA for your protein of interest. And within a certain time frame, your protein will emerge. So that's kind of the view for cell-free in general. But looking at more specifically towards ALiCE, we have two extra factors that you cannot find as standard across other systems. One of these is our functional mitochondria that's still retained in the system, which we use to power the reactions. These powerhouses of the cell here, they're producing ATP energy substrates. And we also have something called microsomes. So these are native organelle-derived vesicle compartments coming primarily from the endoplasmic reticulum and Golgi organelles. And it is these microsomes that really give ALiCE kind of a unique selling point in that you can produce complex proteins. Those proteins that require disulfide bonding and N-glycosylation is really the benefit of using a eukaryotic system like ALiCE in place of something like E. coli, which is kind of prevalent in the literature. So focusing in on those microsomes again, how do we target to our microsomes? It can be explained by the different ALiCE vectors that we use to package our DNA. So if you have a simple protein, which does not require any post-translational modification, like the glycosylation or disulfide bonding, then we have the p-ALiCE1 vector. And these vectors come with our system, with our kit, and you simply can clone in your gene of interest or even have it synthesized directly into the vector with certain gene synthesis providers. And on the right, we have the p-ALiCE2 vector. So using an MSP or a melittin signal peptide is really sufficient to drive your proteins into the microsomes during translation and then have them produced with the disulfide bonding and the glycosylation. Looking at where ALiCE sits and kind of in comparison to other systems. So we have here some reported reference protein yields from cell-free systems derived from various lysates. And here, these different reference proteins are either fluorescent protein or in some cases luciferase, basically simple proteins that you can use to quickly visualize the yield coming out of your system. And the main one factor about ALiCE is our really quite strong yields. So our kit that you can purchase from ALiCE actually has a yield guarantee of 3 milligram per milliliter of our reference protein EYFP, which is quite unique amongst other commercially available cell-free systems. None that I've really seen can offer the same kind of yield guarantee, and that's our full kind of quality control batch release criteria before we can ship out any ALiCE whatsoever. Thinking reference proteins, however, are one thing, but actually useful, potentially therapeutic, difficult to express proteins is quite another. And we've actually done some internal benchmarking of certain therapeutic candidates or gene editing tools internally. So you can see in one column the ALiCE expression yields in milligram per milliliter. And we've contrasted this against expression yields reported in literature for other either cell-based expression systems or the CHO cell-free lysate. And we've come across quite a strong superiority for ALiCE when looking at these certain proteins. So we have here at the top, for example, the BMP2 is a heavily glycosylated protein with disulfide bonds required. So on the very complex end of the spectrum. And we could achieve yields of 1.5 milligram per milliliter in ALiCE in this case, 500 times higher than what was reported for the HEC-based system. Internally as well as in collaboration with our academic partners at the Fraunhofer IME in Germany, we've expressed all kinds of different proteins, a library that's ever expanding. We also have various ALiCE users out there in the world from a kit system. So this is not by no means a complete system. Basically running the gamut from small, simple reference proteins, fluorescent proteins, all the way through to fully formed tetrameric antibodies and membrane proteins. And I will go on to explain a little bit about some of these different cases in the later slides. So that's one aspect, is about the scale, is about the yields and about the versatility of the single system towards all of these different types of proteins. But the other main selling point is that ALiCE is really the only eukaryotic cell-free expression system that has also been shown to be scalable. So moving from the 50 microliter in the first instance is where we do a lot of our screening experiments, expressing a protein for the first time. But then when we're generating larger and larger amounts of protein towards the gram scale, now we can really produce liters of this lysate and also perform lysate reactions of 1 liter. The closest I've seen in the literature for other eukaryotic systems hovers more around 10 milliliter. And in that case, it's a very specialized reactions condition setup. But actually, ALiCE works in a simple batch mode where you simply just add the DNA, and for the lysate, add the DNA and set it to shake for 48 hours, and the proteins emerge. To look at that a little bit deeper, we did some comparison across different scales. We've really found that moving from the microliter plate all the way to milliliters in Erlenmeyer shake flasks, that the yield for these two different reference proteins was exactly the same, regardless of the scale used. You can look into this in a bit more detail in the bioarchive link that's on the slide, but also discloses the reaction conditions for the Erlenmeyer flasks. The idea here is that we want to support people to use ALiCE to be producing their protein at different scales by providing them with the protocols. So the process development is already done, and users don't actually have to do much process development on their own. The milliliters is one thing, but also increasingly of interest now is the liters scale reaction. And this is quite preliminary data, only from an N=1 run of a 1 liter bioreactor system, the Celtainer CT20. And again, we've optimized the parameters on our side so that we can achieve the same lossless yield scalability if you're moving from the microplate to the large scale. So that was an example using a liter scale reaction in the Celtainer CT20 device. But internally, we are also optimizing protocols for a range of commercial off-the-shelf bioreactors that customers or biomanufacturing departments might have available already. The idea really is that LenioBio is shouldering the burden of all this kind of process development so that any eventual ALiCE users can simply just take the protocols, take the lysate from the freezer, add the DNA encoding for the protein of interest, and then within 2 days get their protein out at all these different scales. So with that said, then we really see ALiCE as fitting a niche in various kind of aspects of protein expression. The first instance is the speed of the system. So these are 1 day, 2 day reactions, which getting your protein much more immediately than if you have to worry about upstream seed trains and expansion of transfection and expansion of your producing cells. It's also a versatile system. So the one lysate product, depending on changing out your gene of interest in different DNA vectors, you can produce all these different kinds of proteins from one product. The scalability, as I just showed, is also a really interesting point of our system. No other eukaryotic system, cell-free system, has been able to scale past a milliliter yet without some intense process intensification of the reaction. And these are all work done in a simple batch mode, just for the lysate, add the DNA, and agitate. And with that in mind, it's also an easy to use system. So as I say, we're developing all these protocols, optimizing the parameters so that really everything's available to the user. You just need to ALiCE supply and the DNA and the proteins that can be within reach. So now I'll switch to a few different case studies on what we've done with ALiCE internally. So showing some kind of capabilities of the system with some interesting and difficult to express protein models. So in the leftmost panel, we have some work performed with adalimumab. So this is the blockbusting monoclonal antibody drug Humira, with about $20 billion in revenue in the last year. And so we produced this fully formed tetrameric antibody in ALiCE by expressing a heavy chain and light chain from 2 different vectors. And by purifying out the protein, we could then do some deeper characterization work. So in the first panel, we are looking at some surface plasmon resonance activity assay showing functional binding of the protein that we produced in ALiCE to the TNF alpha ligand that is the target of the antibody. And actually, we saw perfect kind of bioequivalent functionality between adalimumab from ALiCE and from CHO. We also sent these samples off for some N-glycan sequencing data. And we found really reproducible glycosylation profiles, actually, from 3 different batches of adalimumab produced from 3 different batches of ALiCE lysate. You can see there's really tight error bars, show there's reproducible glycosylation across these different batches. This is also quite interesting because no other system, no other eukaryotic cell-free system has yet been able to provide useful amounts of protein to do this kind of in-depth characterization work, but owing to the speed and the yield and the scalability of the ALiCE system is what we could achieve. In the central panel, we see an electron micrograph image from hepatitis B core antigen virus-like particles. I will explain a little bit more in depth in the case study in the following slides, but just to say that we could see these fully formed multimeric structures coming out of ALiCE in the lysate without the need for any additional engineering. And finally, another great example of functional bioequivalence comes from the fibroblast growth factor, FGF, that we produced in ALiCE. Growth factors are of really strong interest right now, considering the cultured meat industry that's ever expanding. Growth factors are needed to culture mammalian cells, for example. And the only current source of growth factors, really, the recombinant ones can be very expensive. Otherwise, you can derive them from animal products, but then that kind of moves away from why you might want to explore like a more vegan cultured meat option in the first place. But you can see based on this bioactivity of cellular stimulation, using the FGF produced from ALiCE, so in the blue and the red lines, we could achieve the same bioequivalence as an FGF standard from Sigma, and also even beating out in efficacy to competitor samples in green and purple. So this is just one growth factor. We've also expressed quite a large library of different growth factors, and it's something that we're really interested in pushing further, this kind of commercialization as well of growth factors produced in our plant-derived cell-free system. So going a little more in depth on the VLP story is that VLPs are really interesting right now in the vaccine space because they are strong, immunogenic, can elicit this immune response in patients. The idea is that the VLP is much easier to produce than a fully competent virus. You don't have to get the package of genetic material in there. You don't have to envelope the virus, which are approaches that would require more cell-based expression. But actually, it's more a purely structural elements coming from a single protein, which then forms dimers and then forms higher order multimeric structures to get these virus-like particles. In the context here, we're using hepatitis B core antigen for expression in ALiCE. So we took the gene from an online database repository, synthesized the gene, and cloned it into our PALICE1 vector. Added that vector to our lysate, and then after shaking the microplate for 48 hours, we could generate some samples sufficiently. So now in the first, we also take it from the microplate. There's our proof of concept expression, the electron micrograph that you saw earlier, using 50 microliter expression, but then also looking at different scales of the VLP production. So moving upwards from 50 microliter through the milliliter scale, using those standard Erlenmeyer shake flasks, all the way up to the bioreactor scale. So this is a 1 liter reaction, again in the Celtainer CT20 device. And we could see that the yield of our protein was really the same across all of these different scales, this lossless scalability, which is really great to show for protein that's not just a reference protein. Moving on to the purification and structural analysis of the virus-like particles. So we could see fully formed virus-like particles in the lysate already. Based on these are molecules of organizations of 180 or 240 protein monomers that form the particles. And so they're quite sizable at scale. You go from the 20 kilodalton weight of the monomer towards these much higher order particles. And so using reverse phase chromatography to purify these particles from the lysate. Having produced our particles from the 1 liter reaction, we could then take it forward to functional studies. So in this context here, we're labeling the virus-like particles using a fluorescent dye, fluorophore. And just by incubating them with dendritic cells, we could see that in the in the negative control, you don't see any fluorescence in the cells after incubation. But in positive control, using flow cytometry and fluorescence microscopy, we could see uptake of the particles. Uptake is one thing, but then actual immune stimulation was also proven. So using these human peripheral blood mononuclear cells that were isolated from different donors, you cultured the cells in the presence of the virus-like particle and then tested the supernatant for production of different cytokines. And we could see quite clearly that we had similar immune stimulation, elicitation, as with the lipopolysaccharide that's used as a control in this type of work. It's also worth noting this low dose of this kind of a vaccine. So here's a 10 microgram per milliliter treatment. And if you consider that we're producing at around 0.5 milligram per milliliter at the liter scale, that is producing half a gram of BLP material. Based on some very, very preliminary calculations, we've considered this to be equivalent to 100,000 doses of a virus-like particle vaccine model within 48 hours in a single liter footprint. So considering the speed at which epidemics can spread and which can become pandemics, using ALiCE as a kind of pandemic preparedness platform is something that we're really interested in. And you can find this work published in Frontiers Immunology just a few weeks ago. So moving from the VLP structure that was a scaffold for potential vaccines towards production of antigens. So like everyone else in the world, we are very interested in working with the SARS-CoV-2 virus and producing proteins of this virus, which can help inform vaccination strategies, can help inform diagnostics also. And you will remember during the boom of the pandemic that everyone, every group, every company in the world had some kind of project working on these proteins. But actually, if you were to try and purchase these proteins, these standards yourself, then the quality could be very variable from different vendors, suppliers, and it's quite difficult to express this protein using a mammalian cell system. I tried it myself and did not manage to get anywhere within several weeks of working on it. But actually turning to ALiCE now, we can show that the receptor binding domain of the SARS-CoV-2 spike protein could be produced quite easily just by within a 48-hour reaction using ALiCE. It's a complex protein, so it's formed a lot of post-translational modifications, which can explain its difficult to express nature. So we have disulfide bonds in there. We also have 2 N-glycosylation sites in the RBD domain. So that's why we'd use our eukaryotic system in place of a bacterial system, for example. Producing RBD in ALiCE was done, of course, in the microscale in the first instance for screening different vector candidates, but then we also moved to the scaled production also. So in this image, you can see what our ALiCE product looks like in the raw form in the lysate that's produced by our system. We can then take this lysate, we simply add the plasmid DNA that encodes for the receptor binding domain in this context, but it could be equally any other protein of interest to you. And then here using a stirred tank reactor to shake the material and produce the protein. Going again to characterization of the protein. So from these larger volumes, we purified the protein in this context just by pure strep affinity. Strep type 2 is located on this fusion protein. And taking it for the glycan analysis, we could again see quite reproducible glycosylation from 3 different batches of the RBD produced from 3 different batches of the lysate. You can see as well here some more plant type glycan structures in orange. This is coming from our plant-derived system. Is quite typical to see. More complex glycosylation engineering projects that we are furthering internally at the moment. But the glycosylation doesn't actually prove to be so important relevantly for the function of the RBD in our hands. So returning to the surface plasmon resonance studies, we could achieve binding to the ACE2 receptor. This is the natural receptor for the coronavirus in humans. And taking that a step further, with the help of a collaborator of Arizona State University, Professor GQ, they also expressed the RBD and some other proteins from the coronavirus in ALiCE, and then took it forward to various kind of antibody-based detection also. And you can see in the left, in comparison to RBD produced from a HEC293 cell line, we could get pretty equivalent binding to different antibodies by producing these antigens in ALiCE. This is taken a step further using patient samples also. So it's kind of serology-based ELISA assay using these antibodies to first bind and then detect the protein. We could really distinguish between infected and non-infected patient samples in blood using the protein produced in ALiCE. This story has also been published in this work on bioRxiv, and the collaborators are still working to develop novel diagnostic formats using the same protein sample, a protein that was really tough to produce in human cells. And the final specific case study I'd like to present is based on the expression of a functional GPCR in our system. So it's quite interesting, the kind of history of cell-free and the landscape of cell-free protein expression in general. We have a lot of people turning to cell-free protein expression for the production of GPCRs and multipass transmembrane proteins. In recombinant protein expression using cells, you're typically overexpressing these proteins so that you can get enough material to work with. But cells really don't like it when you overexpress membrane proteins. It's quite toxic, and not only is it toxic to the cell, but the purification of these proteins is also famously tricky. How to keep these in these membrane-bound lipophilic proteins, how to keep them happy and stable and functional in the various protein purification workflows that are all mainly targeted towards soluble proteins. So as is standard, we had the gene synthesized and cloned into our vector, our PLS vectors. And we tried both PLS1 and PLS2 in this case, using this melatonin signal peptide in case we needed to target two of the microsomes. You remember we have these microsomes that derived from native organelles from the cells. And so we have a kind of native lipid structures already in there. And we found that expression of the GPCR was as a fusion protein with the MBP tag in comparison to how it is expressed in E. coli originally. And we found really high quantity CB2 production in the ALiCE system. This is taken a step further with functional studies performed in lysate, so using a radio-labeled synthetic cannabinoid agonist to study the G protein activation rates in the system. And based on this data, we found functional CB2 activity, which we could approximate to yields of about 0.1, 0.2 milligram per milliliters. And what's really, really interesting about this study is that we did no additional optimization here. If you're working with other systems, you can see in the literature, especially the E. coli systems, then people are supplying all kinds of lipid interfaces, lipid matrices, nanodiscs, detergents, so that the membrane proteins, as they express, have a kind of platform in which to embed. But because we have these native microsomes, we didn't do any need for optimization and could produce this functional GPCR within a 2-day reaction. This is super important for the drug screening industry, for example. These GPCRs are really clear key targets for developing new drugs, but famously tricky to produce, express, purify. So we're kind of offering our system as a one platform to produce these transmembrane proteins for functional screening without any need for optimization. To finalize, I would take a moment to talk about what is the potential for ALiCE as well going forward. So I explained a little bit about our basal high yields in the system. I explained a little bit about the scalability of the system. But another thing that's really why you would choose to use cell-free over maybe a cell-based system is also its use as an open system. So because we have no cell membrane, no cell wall to worry about, you can kind of treat the reaction as more of a chemical reaction, a biochemical reaction, than anything biological. And you can add various chaperones, various small molecules into the reaction to kind of give the conditions that your protein favors best. To engineer this in a cell, we have to also worry about keeping the cell happy, about the biomass production. This is more prohibitive, but in the cell-free system, it's almost living cell-free expression of ALiCE. You can add all kinds of different molecules or expression helpers to get to your protein of interest even better. And the one specific example I would show here is just from some of our kind of design of experiment approaches. So when producing our ALiCE product, we also add the small molecule reaction mix, these kind of salts, nucleotide helpers that allow the reaction to work. And we can show here quite neatly that depending on the different composition of the reaction mix, these are kind of different recipes that are listed on the x-axis here, that we can actually really tune the yield of the proteins that are produced. In this case, it's EYFP here, shown by the relative fluorescence units. But you can see just by tweaking some of the small molecule composition, you can really triple, double, or ablate totally the expression potential in the system. And then taking that forward to a more relevant, more useful application, we can see this data from ALiCE of the VLP produced, the hepatitis B core antigen VLP, where we have seen that if we can, using a novel format of ALiCE, using different format of reaction mix optimized purely specifically for this one protein of interest, we were actually moving from 0.5 milligram per milliliter up to as high as 4 milligram per milliliter. And this is contrasted with EYFP in the yellow bar from a fluorescence read, where we've seen internally anywhere up to 9 milligram per milliliter EYFP2. So we have a one system, our ALiCE product or kit, which is available, that is versatile, optional, open for all kinds of different protein production. But when it comes to the biomanufacturing context, then we might be more interested in performing some additional optimization to really boost the yields of those specific proteins. So finally, the key take-home messages here is that we have the system, the ALiCE system, that can provide access to novel targets and protein classes, these difficult to express proteins with complex disulfide bonding and N-glycosylation, for which you need the eukaryotic system. It's a very fast system, so the idea is to speed up the research and development pipelines from years for new drug modalities up to potentially months. As a cell-free system, using small footprint of liter scale reactions or 10 liter scale reactions at our top end, we can also enable distributed manufacturing. This is a relatively new production paradigm. Example, can you produce a vaccine at the site of an epidemic, as opposed to in one site where it's then shipped out? These are kind of new paradigms that are enabled by cell-free expression. And also we see ALiCE as a great way to look back into the history, look back into the archives of drug production, where there may be some promising protein biologics with great clinical trial efficacy data, but then ran into problems in the manufacturability. Perhaps by switching from the cell-based original platforms towards ALiCE, we can recover some of these shelved products as well. How exactly could you get your hands on some ALiCE to work with ALiCE? So as I mentioned a few times, we have a kit on the market. This can be purchased directly from our website or from distributors. And it comes in various sizes, mini, midi, and maxi, more targeted towards the microscale expression and screening of the protein for research use only purposes. However, when it comes to proteins at scale, or if you are interested in this technology but perhaps don't have the personnel resource to explore this yourself, we also work as a kind of contract research organization with our protein services wing. So if you want to rely on our internal expertise, that's also perfectly possible. And we do these kind of contract bespoke research projects. And when it comes to these larger scale volumes, so the liters more towards the biomanufacturing context, this is quite a new product for the company. It's really the vision of the company was always to move towards the scaling of the system. And now it's very recently achieved. So now it's time to then spread that system out to the world to find interesting therapeutic candidates to develop. And here I'd really encourage people to reach out to us to discuss about these collaboration opportunities as we look for candidates to support as they go towards clinical trials. This can be done via our website, or you can contact the sales team directly via this email on screen. With that, I would like to finish and provide some acknowledgements to the team at LenioBio. So it's quite a large multinational, multicultural team of 40 to 50 people. about 20 of which working in R&D. So any data that R&D manufacturing, so any data that you've seen, it's really been a group effort today. Also the collaborators. So the group of Stefan Schielberg, the original inventors of the tobacco BY2 cell lysate technology before it was commercialized as ALiCE, who we continue to work with. And our collaborator GQ at Arizona State University, who was working on those RBD assays, as I mentioned. The company has received all kinds of funding over the years, so European funding, grants, German government grants. So we have to be very thankful for that also. And for more scientific interest, I would direct you to our bioarchive preprints, which could be accessed via this QR code shown on screen. You can additionally contact us at either of our sites. We have an administrative facility in Düsseldorf on our Technology Innovation Center located in Aachen, both in Germany. And with that, I would close and thank you for your attention.
Thank you, Dr. Williams, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. Let's get started. Our first question is, are there restrictions in protein size that you express with ALiCE?
This is a good question. Not that I've seen. So we've expressed anywhere from peptides of 30, 40 amino acids at the small end, all the way through to monomeric proteins of around 1,000 amino acids. So that really spans the whole range. Also, we talked about multimeric particles, the VLPs, also tetrameric, covalently bound, covalently linked antibodies have been produced in the system. So while there's definitely could be some limitation somewhere, it's not one that I've seen personally yet.
Okay, great, thank you. The next question that we have is, are there any published reports in which authors expressed antibodies using your system?
Yes, so we worked a lot with adalimumab as kind of the globally famous blockbuster drug. That can be found in the preprint on bioarchive, the functional studies, the N-glycan characterization, some disulfide bond characterization also. Antibodies are really kind of interesting points for us. So internally, we've worked with around 10 antibodies or antibody-derived elements such as single chain fragment variables. And all that we've tested have proven to be functional in either in vitro assays or cellular bioassays. So really the system is quite useful for perhaps screening of different antibodies too, where you have large libraries and then you could produce them, sort them perhaps by algorithm towards the higher hitters, and then take those through to the DNA vectors and try expressing them in ALiCE with in-lysate functionality tests is one platform that we're developing.
Wonderful. Thank you, Dr. Williams. Okay. So our third question is, how do you ensure disulfide bond formation, glycosylation, and co-translational membrane integration?
It's quite a range of analyses required there, to be honest. So depending on the protein, really. So a lot of these proteins, their function is intrinsically linked to these modifications. So you could, for example, the case of the cannabinoid 2 receptor is a functional assay which could prove the membrane insertion. But we didn't use, for example, microscopy at that stage. It could be possible to see the insertion. When it comes to the PTMs of disulfide bonding in N-glycosylation, that's where we produce kind of a pure protein at scale and then can send it off for like an in-depth mass spectrometric characterization. But simple Western blot and STS page analysis can also be revealing here. So non-reducing versus reducing, you could see quite nicely the formation of with the presence of the fully formed tetrameric antibodies as opposed to just the heavy chain, light chain subunits. So depending on how deep you want to go, you can use the simple standard assays that are located in labs around the world, or you can go a bit deeper with mass spectrometry.
Okay. Well, it looks like we have time for one more question. Our last question is, can I use my own vectors in the ALiCE expression system? It's
a fair question, because if you imagine switching towards a system and then needing to perhaps transfer your genes into the new vectors, could, depending on how many proteins you're interested in, could be a slight barrier to entry. Internally, we work mostly with the ALiCE vectors, so we can't really guarantee that plasmid vector XYZ will work directly. The important part, I would say, is you need this T7 promoter region, because that is the RNA polymerase that we supplement to our lysates to give it the transcriptional activity. That said, you could also directly supply mRNA. That's the uncoupled version, the purely translational power of the system. But when it comes to other vectors, it would need to be assessed case by case, depending on the T7 promoter, also some regulatory elements in the vector. So for that, I would really encourage people to get in touch with the sales team, the application scientist team, who can provide kind of more bespoke answers to these questions.
Thank you, Dr. Williams. Do you have any final comments for our audience?
So really, I would just urge people to try out ALiCE as a system. It's really undemanding technically. It's a 2-day reaction. All you need to do is shake it in the incubator at 25 degrees Celsius. And like I say, we've expressed so many different types of protein, then I'm quite sure that whatever protein you're working on will come out in the system. Especially if you're having trouble in the cell-based, if it's proving cytotoxic, difficult to express, then really the only thing to do would be to try the ALiCE system. And with that in mind, I encourage people to look into the published reports of how we've been using it, of how other people have been using it, and the range of different proteins that you can get out.
Okay, wonderful. Thank you again, Dr. Williams, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsors, LenioBio, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until April 5th, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.